time, the local time at least, is now 1 o'clock, which means that we can start the proceedings. And I wish you all welcome. My name is the chairman for this event, and my name is here. And because the defense of a thesis in Sweden is a public defense, so anybody is allowed to ask questions, not right now, but later. So if somebody not in the room wants to ask questions, you can send them to me by email, which is written here. So that now what we will have do it. So hopefully you all know that you're here to listen to Ellen Rieloff defending her thesis, which is this one. It's a nice little thick, nice book, and it's sort of titled Coarse-Grained Anatomistic Modeling of Phosphorylated Intrinsically Disordered Proteins. And you will all soon learn what that is all about anyway. So the important people, besides, of course, Ellen, who we defend, is the faculty opponent, Associate Professor Lena Papaleu from the Technical University of Denmark, and also the Danish Cancer Center Research Society Research Center, Copenhagen. So welcome. We're happy to have you here. And we also have a examination committee, which for people in room, we have Professor Ron Fridman from Linnaeus University in Kalmar. We have Associate Professor Per Söderhjelm from the Center for Molecular Protein Science here in Lund. We should have, I hope. Okay, I'll get a nod from there. So we have Associate Professor, Assistant Professor, sorry, Sarah Rauscher for University of Toronto in Canada. So very welcome. I can't see you, but you're supposed to be there. So okay. So very welcome to her, who has gone up very early in the morning to join us. So we thank you for that effort, and we will hopefully hear from her later on. And of course, we also have a deputy member of the examination committee, which is Professor Anders Ehrbeck from the Computational Biology and Biological Physics here in Lund. And obviously, Ellen hasn't done all this. She has also had a supervisor, which is Marie Sjöpe, who will not be able to do very much during this proceedings. Poor thing. Just sit and keeping her fingers crossed, basically. Yes. So that's all the sort of main people for today. So the proceeding will be as follows. First, Elena will give a sort of an introduction to the area, more general introduction, and then Ellen will present her own work. After that, we will have a short break, and then it will be question time, which starts again with Elena, and then the committee will be allowed to ask questions, and then the audience, and including those who are online can then send questions by email. So without further ado, I welcome Elena to the podium.
Thank you very much.
You're welcome.
Okay, I hope you can all see the slide.
We have to share on Zoom.
Right, it's also true. One sec. Then I need to do it like this. It worked before. Just strange like a screen. Need to figure it out in which direction. One sec. Maybe I'll share first with Zoom and then. We should go. Can you see it on Zoom now? So thank you very much for inviting me as an opponent for this PhD defense. I'm enjoyed reading the thesis, and I'm really looking forward to the presentation and the discussions. My duty here is to give an introduction on the topic that we will hear about in detail. So it's going to be a short introduction. I hope I can meet the expectation. I try to think what are the ingredients that we need to know a little bit about to appreciate LN size. And I want to start here with what was the paradigm that for a long time we had in thinking that when we think about proteins, that their structure is strictly intimately connected to the function. If we understand the structure of the protein, we understand the function. And this knowledge started when we started to solve the first crystallographic structure of protein, and we saw regions that were well defined. We have alpha helices, beta strand coming together into a two-dimensional structure. And also another principle that we might want to recall is Amphisan dogma, in which principle that it states that what is in the sequence can predict the structure of the protein. Of course, these things are never easy in reality, and there are much more complex scenarios that challenge this paradigm in this dogma. And if we think, for example, the fact that when we think about the structure in our protein from the unfolded state fold in its structure, it's not only sequence dictating it. In the cell, we have chaperone helping the process. It's not necessary that the structure, that it's the functional structure of the protein in the cell is the more stable. The protein can misfold, form aggregates. And a model we use to understand more in detail the process of the formation of a protein structure is the folding panel model, in which the idea is that the structure, the protein will find its three-dimensional structure going through different states and trying to minimize the conformational free energy. And also here, what is interesting in the bottom of this folding funnel model, we need to think about the structure not as a rigid entity, but a very dynamic entity, in which that bottom, we can maybe look at it as a more rugged free energy landscape with many conformations, and my structure can jump from one to the other. And in doing this, it goes through dynamics, it goes through conformational changes. Another principle that we need to keep in mind to understand the LN thesis. And this ensemble conformation can be heterogeneous, it depends how folded, how rigid the protein is. And the conformational changes, they can span over multiple time scale, depending if we are looking, for example, at small changes in the side chain orientation of the amino acid or more large conformational changes. But then, in the end of the 90s, we started also to have a paradigm shift and starting to appreciate more and more the role of disorder in protein. The role of those regions that are not alpha helices, beta strands, they are not well folded, they're more, and they're breaking the protein rules. And I wanted to recollect this, thinking about this nature editorial I really like, in which there was also a little bit of words from Kate Dunker, that I was the first bioinformatician starting to analyze data in the direction of spotting how much disorder can be important in protein eventually function. And he also said that he has been called many times Mr. Disorder himself, so he fits very well into the into the topic, and I can actually relate quite well with that myself. And I remember at the time I was at the end of my master's studies, so I was pretty young, and I was starting a PhD on another topic, but all this role of disorder was really intriguing for me and something I always cultivated as a side project. And then I ended during the postdoc to work more in this field with simulation myself. Now, when we think about disorder, and we think again to the folding funnel model, then the bottom of the funnel, in this case, it's very flat. So I need to think about this very pliable and a bit chameleonic structure that is unstructured and that can cover multiple conformations. At the point that it could fold in helical conformation, for example, upon binding with an interactor, or he has some propensity to form helical conformation or beta strand or beta small beta sheets. And I need to think about this disorder in very different flavors. We can have a small, almost fully disordered protein. We can have a long region of disorder between domains that are folded by themselves in a more complex and multi-domain proteins. I could have disordered tails at the end of the C-terminal of the protein. And as I said, they are quite chameleonic. They are deprived of a very stable interaction. And this is an example. Is a region of P53 that, depending the one in red or purple, green and orange, that depending on the interactor, so the other protein you need to interact with to exploit, to do its function in the cell, can adjust and change information. And it's exactly the same sequence all the time. And another example is on the other side. But then to add complexity to the complexity, there is another ingredient we need to remember to really appreciate the work that Helen has been doing, is when my protein, when my protein is in the cell, acquire a structure or an unstructured state, there is a type regulation that comes from modification of this protein that are called post-translational modification. And this post-translational modification can be many entities, many different chemical groups can be added to my protein. And one of these that is often exploited by the cell to regulate, tight regulate the function, but also the specificity of the protein is phosphorylation. Phosphorylation is a reversible modification. There are enzymes that are kinases and phosphatases that are deputed to add this chemical group, phosphate, to serine, threonine, and tyrosines, mostly on protein. And kinases are the one adding the modification and maybe activating, or also it could be deactivating the protein, or and the phosphatase will remove it. And it's a tight balance. And this modification will change the nature of our interaction in the protein structure, and the way it could interact with other biomolecules. So it's really important to understand. How we can do this? A good tool to do this is to rely on molecular modeling and simulation. They will allow us to build models that have their own challenges and their own limitations. In looking computational microscope, how the protein can evolve, how is the conformational changes that the protein can have in its native state, how it can be modified by post-translational modification. And an example is, for example, one of the techniques that is presented in this PhD thesis is a classical molecular dynamics, in which we leave the system evolve according to the law of molecular mechanics and integrating the equation of motion, Newton equation of motion. But these models, as I said, they come with limitation. So it's always very good if they are supported by experiments I can do with different technique, different spectroscopy. For example, circular equilibrium has been cited in the thesis, and this is another powerful tool. There are other tools that have been used in this thesis from the experimental point of view that allow you to measure average property of your system. And this, and on the other side, from the ensemble of conformation that I have collected with my simulation, I can calculate the same properties so that I can compare the two of them to understand better my system, its limitation, and how I can also improve my model. And talking about models, So another thing we need to keep in mind is the level of accuracy we use in building up our model. We can span from different models. In this thesis, we are going to focus on all atom models and coarse grain models. All atom, where I treat explicitly all the atom of my protein and my solvent around the protein. And then more coarse grain approach, they are a simplified approach in which each amino acid will be described by bits that can have a certain charge or not. And in some cases, they can become a little bit more complicated. This can be a combination of bits that also give you a little bit of idea of the size. So these models are also very good to address different questions, depending on the time scale I'm interested, but also depending on the complexity of the system. For example, if I'm interested to study the fine detail of the intramolecular interaction, especially upon modification like phosphorylation on a small intrinsically disordered protein, I can rely on all atom. But if I want to study association, there is a nice example in this thesis of self-association of the peptide. The coarse grain approach in the beginning I can learn much more from that, because I can sample a larger conformational space for a larger system. And then I can always come back to the whole atom if I want a deeper description. Yeah, so with this, I think I'm done. I hope that this gives you a little bit of idea what you will hear about. So we're going to talk about modeling and simulation and experiments, how they can come together, understanding protein that are disordered in their structure, understanding the impact of post-translational modification with focus on the phosphorylation, and appreciating the potential and the challenges in using coarse grain models and all atom models. And I will leave the space to Alan.
Thank you very much.
And then I will just take some water, and then I will start. So thank you for being here today, and thank you, Lena, for a nice introduction. So as you heard, my thesis has the title Coarse-Grained Anatomistic Modeling of Phosphorylated Intrinsically Disordered Proteins. And then to understand intrinsically disordered proteins, then I gave an introduction, but we start again with the most basic level. Proteins are made up of amino acids connected into long chains. We have the generic structure of an amino acid here. So there are approximately 20 of them, and they all share the same basic structure, but then they have different side chains. And depending on the side chain, they can have they get different properties. So they can be classified as polar, hydrophobic, positively charged, or negatively charged. And the order in which these amino acids are connected is called primary structure. And that is illustrated here. And then depending on how the primary structure look, it can fold, the chain can fold, local parts can fold into more defined structures. For example, alpha helices and beta sheets, which is referred to as secondary structure. And then the full chain can also fold into a well-defined tertiary structure. So a well-defined three-dimensional structure. And as Elena said, for a long time it was believed that this three-dimensional tertiary structure defined the function of a protein. But this was contradicted by the intrinsically disordered proteins, as they actually lack well-defined tertiary structure. And this makes them much more flexible, and they have a rapid interchange of different conformations. So in one moment they can be more stretched out, and then they can be more folded over and interchanged like that. Despite this, and also thanks to this, they are still biologically relevant and involved in processes such as signaling and regulation and recognition. And they also are associated with several human diseases, for example, Alzheimer's and Parkinson's disease, and different types of cancer and diabetes. So there's a large interest in understanding how these proteins work, but they are more challenging to study experimentally due to interchanging structure so much. So the other key word in my title is phosphorylated. And as Elena introduced us to, the phosphorylation is the type of post-translational modification, and it is employed by the cells as a regulatory mechanism. And here's an example of serine. When it is phosphorylated, you see we have this extra phosphoryl group, which then adds more negative charge, and also enables the formation of hydrogen bonds. And it also makes the amino acid larger, so more bulky. So this, of course, then can affect the structure of the protein, how it interacts with other proteins and constituents, and then how it behaves, the dynamics. So my research has been focused on contributing to the development of computational tools for study IDPs as a complement to experimental methods, since that is challenging, and then also to increase the molecular understanding of IDPs, especially focusing on this relation between the sequence and the structure and how phosphorylation affects it. So in my work, I've used different methods. Modeling and simulations are the computational methods, and then the experimental methods I've used small angle X-ray scattering performed at the European Synchrotron Radiation Facility in Grenoble, and then circular dichroism as well. And in small angle X-ray scattering, SAX for short, with that method we can obtain the average size and shape of the protein. So here is an example of a form factor measured, a form factor from SAX for a globular protein, more Gaussian, which is how most IDPs behave, and a more rod-like protein. And we can see that they look different here in these representations. And especially here in the Kratky plot, we get a nice indication of the shape of the protein, since the globular one has a more bell-shaped curve, the Gaussian one reaches a plateau, and the rod-like one more steeply rises. And we can also determine the average size through the radius of gyration. And the other method I've used is circular dichroism, from which we can obtain the average secondary structure content. So this here is an example of CD spectra for proteins of different secondary structure. So you can see that the spectra look really different, and in that way we can see what the average structure content is. But as I've said, we get average information from the experiments. And when you have an intrinsically disordered protein that changes conformation a lot, then the average doesn't always say that much. So therefore, we want to have these simulations as well to give more details at the molecular level. So here we can more easily see special interactions and what is happening over time. So the simulations can help us with interpreting the experiments, and they can also be used to predict what is happening. Experiments can be very time-consuming and expensive, so then it can be nice to try it out with simulations first. So why don't we always use only simulations? Because to perform simulations, we need to have a model, and we need to know that this model is accurate and work for the system. So then we need to compare it to the experiment. And then also some systems can be complicated and complex and just out of reach for for simulations. So Elena gave us a nice introduction to how modeling can be performed in different ways. So this is an alternative representation of this. Here we have a cow that we want to model. And we can have a more detailed model, as you see here, or we can make it more coarse-grained. And in the end, it doesn't look anything as a cow at all. So we need to find a suitable level where we can get out the information that we want. The advantages of it being in something more coarse-grained is that we get a simpler model with fewer parameters, so it is easier to interpret and understand. And it is also faster to simulate, so we can study larger systems. But we lose detail, so we might lose information that we need. Also, with the surroundings, we can simplify it by representing the salt and the water molecules implicitly instead of having them explicitly there. So I've used two different types of models. A coarse-grained model, where each amino acid is modeled as a hard sphere, and those are connected through harmonic bonds. And each sphere has a charge, positive, negative, or neutral, depending on the amino acid residue. So we take into account the electrostatic interactions and also have a short range attraction to model an approximate van der Waals interaction and use implicit solvent and salt. And then I've also used a type of atomistic model where then each atom is explicitly included, also the water and ions. So this model has more potentials, as you can see here, and also a lot more parameters. And together, the potentials and parameters that define the atomistic model is usually called a force field. And there is continuous development of different force fields. In the beginning, a problem with force fields for intrinsically disordered proteins were that they first give a lot of bias in secondary structure and also yielded much more collapsed structures. But now we have better force fields available. So now to my papers. So in the first paper, we worked with this coarse-grained model that was parameterized for a saliva protein histatin 5. And we wanted to see if this coarse-grained model also can describe other IDPs, so if it could work more generally. So we performed Monte Carlo simulations of a single chain using this coarse-grained model of 10 different intrinsically disordered proteins. And they differed in length from 12 to 248 amino acids. And they also differed in proline content, which is interesting because a higher proline content usually corresponds to a more stiff protein. And they also differed in hydrophobic content. And this figure here is a diagram of state, where these different fields represent different conformational classes. So the proteins that fall in this area, they are predicted to be more globule-like, and the ones here are predicted to form more coil-like structures or hairpins. This is an intermediate region, and here we have more extended structures. So you can see that these 10 IDPs that we have studied fall into different categories. So they represent a range, even though they are not that many. So here we have plotted the radius of duration determined from simulations against it determined in experiments from SACs. And when they match perfectly, they fall upon this gray line here. And you can see that overall, we have a rather good correspondence between the simulations and the model, the simulations and the experiment. There are some deviations. For example, these yellow ones are the proline-rich proteins, and they are a bit smaller in the simulations, which is expected since the simulations, the model doesn't take into account these prolines. And then also the highly phosphorylated ones, these two red ones, are also smaller in the simulations. So overall, the agreement was rather good, but there are some weak spots. In the second paper, we continued the work with this model and wanted to apply it to a more complex system. So here we studied self-association and wanted to know if the coarse-grained model could describe this and also help us understand what governed this process. So here we use staphyrin, a saliva protein, as the model system. And here we have the primary sequence. And you can see that it has a distinct charge pattern. So here is a block of negatively charged residues, including 2 phosphorylated serines. And then we have a block of positively charged residues. And then the rest of the chain is mostly uncharged. And we used SACs and Monte Carlo simulations using this coarse-grained model, but with an extra attraction to induce self-association. And here we have some of the SACs results, where we can see that when the protein concentration is increased, this intercept with the y-axis increases. So that signalizes an increase in molecular weight, so that we have a self-association in the system. So we can, from this molecular weight, we can calculate the average association number. And here it is plotted against the protein concentration. And we have a rather linear rise here in this concentration regime from the experiments. And the simulations show the same here in the beginning. But then the model breaks down and everything aggregates into pretty much one large complex. And that was shown to depend on the use of implicit salt. When we had the salt explicit instead, we had a average association number more in agreement with the experimental data. So as long as we are in this lower concentration regime, this model seemed applicable. So to understand what governs the process, we needed to see how it responded to changes in different factors. So first, we saw the protein concentration. An increase in protein concentration gives an increased average association number, and that was also shown in the model. When we added urea to the system, the complexes became smaller. And urea is known to weaken hydrophobic interactions. So this indicates that the process is hydrophobically driven. And then when we added more salt to the system, the complexes grow larger. And when we add salt, we screen away more of the electrostatic interactions. The electrostatic interactions are also important in balancing the size and stopping the self-association. And then when we increased the temperature, the complexes became smaller. And when the temperature is increased, the entropy becomes more important in the system, the chain entropy. And also we saw the same thing in the simulations as in the experiments. So to conclude, this indicated that the process, the stuttering self-association, is hydrophobically driven, but it is balanced by the electrostatic interactions and entropy. And the model was able to qualitatively capture these effects. Then for the last 3 papers, we switched focus to these phosphorylated residues and wanted to see what conformational and structural changes are induced upon phosphorylation. And then to see more the secondary structure, we needed a more detailed model. So then we switched to atomistic simulations. And we want to see if current force field could work for these phosphorylated IDPs. So here we started to study only the first 15 residue of stutterine. So this part where almost all charges are located. And we used 2 different force fields that have been shown to work well for short non-phosphorylated IDPs. So we measured SAX on this, and here you can see the form factor, and they look almost indistinguishable. The phosphorylated and non-phosphorylated variant. And here in the Kratky plot, we also see no real difference, but also the uncertainty in the phosphorylated data is rather large. But a fit to the data suggests that we might have a small contraction. It is plausible. In the CD data, we see a larger effect. So here we see the changes in the CD spectra, which indicates an increase of alpha helices and beta strands. Then here in the simulations, if we first start to look only at the non-phosphorylated peptide, these blue lines, we can see that both force fields are in agreement and nicely matches with the experimental data. Then the phosphorylated, on the other hand, we see that the force fields give different results. And then also we see a compaction upon phosphorylation. So there's a larger change going from non-phosphorylated to phosphorylated state. And then when it comes to the secondary structure, here we have the propensity for helix plotted against the residue index. So for the amber force field, when we go from non-phosphorylated to phosphorylated, we see an increase in helix propensity in this first region. But then in the charm force field, that doesn't happen. Instead, we see a decrease here in the middle region. What we saw in the charm simulations was that we got a lot of this conformation where the phosphorylated serines form salt bridges with the arginine residues. So strong salt bridges that they kept the peptide together in this bent conformation. So in the fourth paper, we want to see if this only happened in this peptide or if it was a more general effect. So we extended the study with 4 other phosphorylated peptides. As you can see here, they differ in length and they have a different charge pattern and also a different number of phosphorylated residues. And we performed atomistic simulations using the same force fields. And here is the radius of gyration, the distribution of radius of gyration. So the blue curve is the amber force field, and the orange one is the charm force field. And we can see that for all 4 peptides, the charm force field yields more compact conformations. And we could see that here is the probability of forming salt bridges between the phosphorylated residues and the positively charged residues. And once again, the orange corresponds to the charm force field. And we can see that the probability is much higher in the charm force field. So there we have more salt bridges, which explains the more compact conformations. Then in the last paper, paper 5, which now is also published. When I printed the thesis, it was only submitted, but now it's accepted and published. And here we wanted to see the conformational structural changes that are induced upon phosphorylation. I want to see if we could see any patterns or see what factors controlled it. So then the study was extended with also the non-phosphorylated variants. And here we only used the amber force field, since compared to experimental data, it appeared to perform better than the charm force field. And we can see here, once again, it's the distribution of radius of gyration. And as you can see, it changes upon phosphorylation. And it was recently hypothesized that the induced changes in global dimensions could be predicted by the net charge of the non-phosphorylated peptide. So for these 5 short peptides, we have here the change radius of gyration and the net charge, and we see no relation between them. Now I will only show, go into more detail about 2 of these peptides. So for this beta-casein fragment, here is the energy landscape. So in the non-phosphorylated variant, we see that the mostly visited conformations are of this type, rather extended. But then after phosphorylation, it instead shifts to be over here with a bit more compact structures. And we can see that in all these conformations, we have the positively charged N-terminal close to this phosphorylated region. So we saw that this peptide contracts due to the electrostatic attraction between the positively charged and residues and the phosphorylated residues. So this really shows the importance of the charge pattern. And then for statuarine, we saw that it expanded upon phosphorylation. And here's the propensity for beta strands, another type of secondary structure. And we can see that in the non-phosphorylated variant, we have these sharp peaks here that then disappears after phosphorylation. And here is an example structure showing how these beta strands and beta bridges keep the peptide in a more compact conformation. And here is this residue here is an arginine, and over here we have the tyrosines. So we can see that the arginine and the tyrosine interact in the cation pi interaction, and then it greatly diminishes upon phosphorylation. Instead, we see that this arginine involves in a salt bridge with the phosphorylated residues instead. And then these beta strands disappear, and the peptide can be in a more extended conformation. So to conclude, we've seen that the coarse-grained model seems to be applicable to many IDPs. And when it comes to the phosphorylated IDPs, the stability of salt bridges is critical for obtaining good results from atomistic simulations. And we also saw that it is not straightforward to understand how phosphorylation impacts the structural and conformation. There can be different effects on the local and the global level. And also the charge pattern matters. The presence of aromatic residues mattered. We saw for the tyrosines and saccharin. And the chain length, the number of phosphorylated residues. So yeah, there are a lot of factors, and they can interact in different ways, so it's hard to predict the outcome. So to continue this work, it will be interesting to study the charge of the phosphorylated residues in the coarse-grained model and also in the atomistic model. It is a fixed charge of -2 for the whole residue together. And perhaps that is not the best representation compared to what is happening in nature. And also to improve the force fields, this stability of the salt bridges is important. And for further validating force fields, we need to have more experimental studies with more detailed information to compare with. And here, NMR experiments where chemical shifts and decouplings and NOEs can provide more detailed information that can be used then to validate the force fields. And we're also seeing that the experiments and the simulations really go hand in hand if we want to understand this intrinsically disordered proteins. We need both parts. So last, I would like to thank my supervisor, Marie Scheppe, and then also my collaborators and people who have helped out at the Beamline. Yeah, and lastly, you for listening. Thank you.
Helen. So yes, we will now have a short break. We'll move into the frame. So let's say that we'll be back at 2 o'clock. So on the hour. Let's big stretch up. Welcome back. Welcome back, everybody. So now we will proceed with questions time, and I'll give the word to Helena.
Thank you very much. So first of all, I want to congratulate with you for this nice thesis, your very nice presentation. You did a great job. And I really enjoyed it. I have to say that I appreciated, because I think you kept a very good balance in the introduction, in the methodological aspects, and also because you have the challenge of presenting both the simulation part, the computational part, and the experimental one, to give to your reader all the pieces of information that are really needed to understand what is happening in the publication, in the part of the manuscript. I have just more notes so we can go around. And also I like to see the evolution that you had in your thesis, in both sides, like from the scientific point of view, the fact that you started with work with a coarse grain model and then building up on complexity of the question you wanted to ask, or complexity in the model that you wanted to apply. That was really, really nice. And also that you had with you a good model system that it's sort of the union point, the paper, but then you were also exploring other protein with your techniques, and that make it very, very solid. But then also the evolution that I have to say I can see in you building up in independence in the work, starting with the work in which you were one of the co-authors, starting to get adjusted to that, and then first starting to take ownership and also leading the publication. Really, really nice. But of course, I have to ask some questions, and I think the way I'll do it, I have some notes that I took when I was reading, so I go through them. So we probably sort of follow the order of the thesis, but then I have some points that, I think I will tackle them during the discussion. I was inspired during the presentation to ask. So starting from the introduction. I was a little bit with mixed feelings when you said, and it's just the first page of introduction, that IDP, yes, they lack well-defined structure. They have proven more challenging to study experimentally than conventional protein. But I don't know if I like very much the word conventional protein.
Yeah.
But also is really true, because if we think about what we consider folded protein, they might be challenging as well.
Yes.
What do you think? I mean, there may be a lot of.
So what I meant about it was this with, as I said in the presentation, that they don't have a well-defined structure. So when you get an average, you don't get that much information. But of course, like performing the experiments can be tricky, and the proteins, the globular proteins can be sensitive in different ways as well.
They have a lot of dynamics also going, and very heterogeneous.
Yeah, and they aren't that rigid either. And
then often it's a combination of disordered and folded protein. And then a little bit later, when you say that simulation are useful for making prediction and investigation and condition unattainable by experimental method. Maybe you want to comment a bit on this. What did you mean with this unattainable? Is it because it is related to what you were saying in your presentation, that in a way experimentally we have average property, and then with simulation we go at the level of the individual structure, or what did you mean with this?
And for example, we can take stacking as an example, as it self-associates. If you want to see only how the single chain behave, but then you need to be at a certain concentration to be able to measure with stacks, for example, to get the useful signal. But then it self-associates, but in simulations you can see just the single. You can study.
I guess also experimentally it's something you can try to do for single molecule based approaches. I was a bit also mixed feeling, in the sense that, at least, and this might be just my personal opinion, I want to share. I think it's really important we try to really go hand by hand, as you said, so that when I simulate, I try to simulate something that then I can, even if challenging, measure experimentally. Not that I try to simulate something that I will never be able to achieve experimentally, because then, as you said, so maybe it's a bit of the wording here. I'm not going to be so capable of evaluate the accuracy that I have in my model, and how I can make it to that.
Yeah. First, you need to evaluate the model, and then if it seems really solid, perhaps you can go a bit outside the range. Yeah, and get some more information.
Yeah. Then we have the part more, let's say the introduction to the IDP, what they really are. And then you say, page 5, that at least 10% of the cardiac protein are intrinsically disordered. I think this number is probably more now.
Yeah, and probably.
But I don't know how you were estimating this, if it's we are talking about almost fully disordered protein or disordered.
Yeah, I meant the fully disordered, because when you look at these proteins that have long disordered regions of like 50 residues or more, then you're up to 30% and more. Because if you give yourself the criteria like that in your protein, you're okay with seeing 30% of the disorder overall. Then it's more than 30% of the cases in eukaryotic cell. And they are also often the ones that are, as you mentioned, very important also in the context of cellular function and disease, because they often play in the crosstalk between interaction and pathway. And then you mentioned that it's nice thing to mention that many IDP have been shown to have folding induced upon binding interaction with the partner. So now, if we think about protein-protein interaction, when at least one of the two players is an IDP, it can also be between two IDP, right? Can you think about other mechanisms? Because it's not necessarily that folding upon binding is the only one for molecular recognition of an IDP. What else could happen there? So I didn't fully understand the
so when we think about the mechanism of interaction, of recognition between an IDP and another protein, or two IDP, you are mentioning this folding upon binding. So I have a very disordered protein, I recognize my partner, and I use it to stabilize the structure. Some of the examples I showed, they were folding upon binding, right? But there are other mechanisms for the protein to recognize. This is just one of the examples. It was the classical example. Can you think about something else?
It can also be like special motifs that they recognize in each other, and it doesn't need to be a change in structure upon that.
So you're saying they can stay disordered, the binds and stay disordered. That's very much it. They can also stay very disordered and with the motif that maybe it's a bit rigid, or I can be a bit more fuzzy and dynamics. They can still fold. They can still be folded when they bind, but then it's not really induced by the binding. It's more an intrinsic property of the chain of the protein that occasionally will sample have propensity to a certain state, like an alpha helix, and this gets stabilized by the interaction. Something around the conformational selection, if you want to call it that way. So it's really it's really diverse the way they engage in protein-protein interaction. Then for the classification of IDP, you are focusing a lot on the dust and PAPU diagram. That it's really useful to understand the property, especially when this is triggered by charges. And it's very important for your study. But Are there other models that one could use also to understand, for example, the impact of hydrophobicity or lack of hydrophobicity, or also this proline that you mentioned? Can you think about some other models that have been developed? I think for a certain extent using this in some of your publication, because you don't only look at the charge distribution, but you are also monitoring what it changed in terms of proline.
Yeah, because the hydrophobicity is important, and also for like compared to globular proteins, to differentiate between globular and intrinsically disordered. And then
what many prolines could give in a protein that is isolated?
Usually it's one of these more disorder promoting residues.
No.
Prolines are more common in intrinsically disordered proteins than in globular ones. I
because
believe.
You can think about them as helix breaker, but sometimes if they come in the combination with other residues that can form this. You mentioned a limit.
You can also
particular secondary structure.
Yeah, this PP2 structure.
Polyproline.
Yeah,
but it's rather unstable. So borderline. So and you have some of them in your sequence, so that's of course a property to look into. And there are other features at the sequence level that we came to understand better in the last year, because you mentioned the process that IDP can go through, that is liquid phase separation. So there, I think, there are some preferences toward certain amino acid composition.
Yeah, and you often have like a low sequence complexity, so not that many different amino acids, and then like motifs that can be repeated, and for liquids phase separation, you can have these weakly associated.
And from the point of view of the interaction, of the weak interaction, which one is the special interaction that is very often occurring in this separation field? So we are only looking at charges here, but what about aromatic?
The tyrosines are often involved in the liquid phase separation, as far as I know.
And it was striking me that your sequence has all this tyrosine around. Is it known if we can undergo phase separation?
I haven't seen it, but it would be interesting to look more into statuine. It seems that statuine can be studied from many different points of view.
Yeah. And so you, out of curiosity, when there are some predictor of could tell you the potential to go through that business sequence that you could try out. So this was no, I don't know. Then in the introduction to bring closer to your model system, then you were talking a bit about the environment in the saliva, and that this is just my curiosity. Is it because you said that there are many of these proteins that are intrinsically disordered in the saliva as an environment? Even if it's a fluid that doesn't have super many protein, maybe. I don't know if there is an estimation of actually the percentage. So if you take I would say the protein into the saliva, how many of them are intrinsically disordered? And it's actually
Oh, that I
more
don't know.
more than what we see in the entire. If you look at the distribution in the entire. Body.
Yeah, that I haven't looked into. So
Yeah. It was
I
a
don't know.
curiosity.
Yeah.
And then we come to the stuttering. I know almost nothing about this protein, so maybe what I'm going to ask is a bit naive or unknown, but it was very interesting to read about it. You said that it prevents spontaneous precipitation of calcium phosphate as a salting as a function. But is it known if you can also directly interact with the calcium, for example, could chelate the acid agent? No.
Yes, you can.
You can
Through this acidic
uh,
motif with the
yeah.
phosphorylated residues and the other acidic residues.
Any of your colleagues tried to model actually the interaction with the calcium and look in the phosphorylation output affect that?
No, no, we tried to do Sachs measurements and see if we saw any changes with adding calcium. But then it was with increasing the protein concentration, we fast reached a level where it was just aggregating, so we got these like milk white samples. And just keeping it below that level, we couldn't see anything with the insects, but it is the low resolution technique. So then we didn't continue looking more into that, but
and then of course you might have some challenges also modeling wise with methylene, but not so many things for calcium is still in a regime we can say something meaningful. This one I asked. Then another curiosity of this party, because when I was reading your thesis, then I just went to UniProt, which is the data bank for the sequence. I was looking for the protein, and it's reported as a longer sequence than this one, with something in the N-terminal, because you say that it's a 43 residue, but there it's reported 62. I was actually wondering what this part that it's N-terminal is because it got cleaved or?
Yes, and I don't really know what that part is. I have just accepted that, this is the active.
The active part,
yeah.
So maybe some processing of the chain to release some cleavage event. Yeah, that's funny because that part seems to be much more structured, like so AlphaFold predicted as a helix, I think. Yeah, I was wondering if it was because of some cleavage event, or maybe it's actually there may be different isoform, and this one is the one that is more expressive and it's short. And then we move to I want also just to have time also to go to the results. So let's see if I can. I think the part in which you described the different contribution to intra-intermolecular interaction is really nice. Maybe here is where I could say maybe as a reader I could have appreciated to read a little bit more about conformational entropy. Yeah, I felt like, okay, we are discussing so much about the enthalpy contribution, and then I reached the end.
Sure.
Maybe you want to comment a bit more on this part, and also how important this could actually be for IDP.
So like entropy is important since the nature strives for like disorder and more possibilities. So and like when an IDP, for example, encounters a surface, then it gets restricted. It cannot adopt that many different conformations as well. So it is not favorable to be there. And the same, of course, when it self-associates or induce a fold. It also loses conformational entropy. So it is a part of this balance with the other energetic contributions.
That will balance out. Then we have the simulation part with the different models. I wanted maybe to discuss some reflection on your side, because you have been using this coarse grain model that is really coarse grain, because we are narrowing it down to 1 bits and charges or not charges. There are other coarse grain model you could explore, and now that you have seen both the side, because you have seen this very minimalistic coarse grain model and then what you could think about for your system or for the system you have studied, a coarse grain model that can introduce some property you lose here, but without going totally all atom, what you will do? It's true that, I mean, if you look, phosphorylation charges are going to have a big contribution. We are putting this, assuming to put this -2. But it's only the charger. What also a phosphorylated residue bring to the structure?
Yes, so the possibility for hydrogen bonds, and also, the size changes.
The steric hindrance.
The steric hindrance.
And then you have the phosphorylated.
And there you have an alternative model, like more accounting for the steric hindrance with different sizes of the amino acids.
Different sizes also of the beads, or at least,
or adding like a second bead for the side chain.
Okay, then there was a statement I'm not really sure I understand. It was the one, there is a section on the force field.
Which page?
27, in the very end, last paragraph. That you are discussing a bit about the fact that, of course, I mean, we have optimized force field for certain systems, so they will work better for them and for something that is similar to that. And then you say this also extended to the type of properties considered for validation. But I don't know. Maybe it's just me, but I don't understand what do you mean with this.
Like if you optimize it after like NMR data focusing on secondary structure, that doesn't need to be related to like the global conformation that you can see in SACS, for example.
Okay. Yes, so different experimental approaches will tell about different properties.
Yes, and the global and local structure isn't always connected.
Then some more into some choices that you made in terms of running simulation, that you use several replicates where, if I understand correctly, the starting structure is the same, but then we are changing the random seed to the get. So every time you get associated to different velocity, right? Could you think about other way to actually try to increase the sampling without using this trick, so to do the replicates in another way, especially because it is an IDP. So I guess your starting model is just extended.
Yes, it is. I'm not really sure I understand your question.
Because so one way to have more than one simulation for the same system, and then you concatenate them. One way it's one to change the random seed, but then you just change.
We can change the starting structure as well.
I think so.
And
because then it will give you maybe a better coverage. If your problem is also the coverage of the conformational space here.
Yeah.
Because you want to try to end all the time in different places and then do an event. Of course, it's tricky to think how to do it because you might induce some interaction that you don't know already in the starting model. So it's a bit tricky, but that could also be another way. And then how did you decide for I don't remember now exactly, but I think in your studies you were using something like 5 replicates per system or something like that. Is this based on some previous experience with
yes. So the people working with atomistic simulations in the group before me did it in that way, and it worked well, and then I continued.
And since we are talking about the starting structure, that you're starting with this very extended one, then I was a bit curious. You built, and you are using a water box around the plotting. That I read it's cubic. No, it's one of the.
It's rhombic.
Yeah, and it's also nice that in the introduction, in the methodological introduction, you explained why it's a good start. This start of the cubic one, but so this will give a very large box, right? So how do you decide to check for issue like periodic images looking?
So I look at the minimum distance between the protein and its closest mirror image, and compare it to the cutoff of my interactions.
And did you have did you design the box with the proper dimension, or you did some tests trying to increase them just to find maybe a balance between not having too many atoms?
Since the starting structure is fully extended, then the box is rather large to fit that that extended structure. So then when it usually folds up, it's no problem. But then of course
became a bit slow to simulate. So
a more efficient way would be to maybe do a test run and then use a different box size to get rid of some water.
Yeah, we have the same challenge. We do this. Yeah. Okay, I think I will just skip some of this just to go to the results. You have a lot in your work, a lot of PCA plot that you use that are, if I understood correctly, they're all done in the Cartesian. Yes, right, with the label. That's really nice for IDP because then associate with the different propensity to conformation. And then you try to estimate the sort of free energy, informational free energy, conditional free energy out of it. If you would have more time and more resources, would have you explored other approaches to calculate this free energy landscape? And which one maybe you read about, or you, let's say you still have some years on this project and you want to try something else because now you think, okay, but I only did classical MD and I'm a bit bored, I want to try something else. What you would try?
Now I can't remember the name of methods, but there are other methods for like obtaining more the potential mean force and and I think that would be interesting in this system.
Yeah, you could rely on what we broadly call enhanced sampling.
Yes.
Which, for example, could define some reaction coordinate or collective variable that still tell you. So your 2 points are how compact the protein can be or not compact. So you could play around properties like that, and the residual secondary structure, if any. So you could design, and actually to have already the knowledge of how you behave in the classical MD help you to design that step, because you can use your knowledge here and try to also further test some hypotheses. Also, what these differences you see in the force field will still be there if I enlarge my sampling, or maybe it was also because I was just sampling that was scattered away different parts.
I think different enhanced sampling methods would really be good for these systems.
Yeah, I think it would be really nice because it's not too big. I'll leave the questions to someone else. One thing that you said in the introduction is that saliva is an environment that it's very sensitive to changes, especially of pH. Then I was wondering, do you think for your system, well, I couldn't say now, so what it's the change there, how much the pH can change, but for your system could be interesting also to try to play around the pH in your modeling, or it doesn't make much sense?
It could be interesting, but then experimentally we had troubles with exploring different pH due to this self-associating properties and aggregation. Then when you change the pH and it becomes less charged, then it aggregates much easier.
Yeah.
But also for these like phosphorylated residues, the pH is important to consider.
Yeah, absolutely. Then, maybe it means one should also think about what is the shift in pH that could be meaningful to simulate, because well, at least a good thing for you is that your sequence doesn't seem to have any histidine that generally are super sensitive to that. So it's not necessary having to overcomplicate your life with modeling. Already is complicated. Then for the self-association, in the introduction, the self-association study, in the introduction, you presented as an example that has been already studied, and for inspiration, the data casein, right? That lot was done. I was wondering if it could make sense to bring it with you as because we are very much interested in comparing force field and approaches. Bring it with you as another model system.
Beta casein is much larger.
Okay.
It's closer to 200 residues.
Okay.
So
therefore,
not. Then we would just have to make it study it in a coarse-grained
model. Yeah, in a coarse-grained model.
But it's an interesting system, and in beta casein, the temperature effect on how temperature affects the self-association is the opposite of what we see in stutterine. So it's interesting to compare the two, and it shows that it is
in the studies they always worked on the full length beta casein, so it doesn't probably. I don't know. I was wondering, they never tried with smaller constructs that could still be a minimalistic model.
Not that I've To my knowledge, for the self-association of beta casein, you need this long hydrophobic tail.
But then maybe for the cross-grain part, it's also if you want to develop your model further, you can bring that with you. Then another very interesting discussion is the fact that when you compare for the phosphorylated variant, Charma 36 and Amber, faster than the 99, you have this different behavior. One of them seems to increase rhelicity, the other one is decreasing, and then you have this tight arginine phosphorylated residue interaction. And then there is the discussion on the fact that we need to be careful with force field that they overestimate. So the bridges that I really like it, and it's also something that is a bit of my obsession as well. But this maybe it's something I lost. Maybe, so when we have the not phosphorylated variant, you still see this tendency of one of the two to have a small helical propensity compared to the other when you sample. I was thinking how much is a problem with force field in general, also for not phosphorylated protein.
Or they were pretty similar in the non-phosphorylated state. There were some differences in secondary structure. I don't remember exactly now. I will look at the plot.
Because if you cannot make it from
so there were some, a little bit more helical structure in the amber force field than in the charm force field for this SN15 peptide.
Then it's something in the phosphorylation, and then you have this hypothesis that so in general. Maybe one problem is, if understood correctly, is the fact that we are modeling it as -2. So we might need to actually play a bit around the charges there. And then this general problem of bridges that are overestimated. But it was also attracting my attention a lot that what you have in the phosphorylated version is this cation pi interaction, right? If I understood correctly, when it's not phosphorylated, you have the cation pi interaction.
Yes, in statuary we saw that.
And tyrosine.
Yeah.
And then you get the phosphorylation, and this seems to be replaced by the salt bridge. Could we look at the problem from another point of view, that it's not only a problem of overstabilizing salt bridge interaction, but how these forces deal with interaction that have an aromatic component?
Are
they good or not?
That I haven't really looked into, but that is an important point.
So I think there is also where we need to think about a bit in the field then, because it's maybe a combination of the two things.
And I would also like to see if we see these effects experimentally as well.
That's what I want to because now you have really this. Is it something that is a state that exists with this interaction or not? Or is it really a limitation of force field?
At least I did some CD measurements of statuarine as well, and non-phosphorylated and phosphorylated variant. It's not in the papers, but then we saw this decrease of beta strands. So something is happening also experimentally. But then to really compare it, we need to have more detailed methods.
And also maybe that starts to be an interesting question for an MR, I would say, to address.
But something we wanted to, if I had more time, we would move into NMR.
You can leave it to some future students inspired by your work. Then when we think about phosphorylation in a protein, that seems to have this propensity to helical formation, at least according to what you agree on. That seems to be there because you have it in the CD and you have it in the simulation with Amber. There is an interesting structural mechanism for the phosphorylation in stabilizing helical conformation that you mentioned that depend where the phosphorylation is, if it's doing this capping in the N-terminal or somewhere else. Because phosphorylation on helices, they can do a lot of things. They can stabilize it, or they can actually destabilize it and decrease the potential. In your case, did you see what did you see? This phosphorylation, they tend to be when the helix is formed N-terminal, or you have this capping mechanism?
Yes, I also saw this distance to a positively charged residue, a lysine or an arginine. It contributed to stabilize
so that would be the lysine that you have after the 2-glutamate.
Yes.
Yeah, so you had that interaction. That's pretty cool. We have a short linear motif that behave like this about phosphorylation. So I would be curious to compare to what it was a publication we had some time ago. So I don't remember now the doctrine, but because we are still trying to figure out. I mean, we see this feature, but we don't know what does it mean exactly for the structure, for the function of the protein we were studying. So see it again on another case. It's very interesting. Did you look into this polyproline propensity?
Yes, I did.
And
yeah,
substance.
but like I think all the peptides that I studied had a significant part of polyproline structure. But then, like changes in it upon phosphorylation, I couldn't really see any trends, or in some cases, nothing happened to it as well. But perhaps it's important in like just when a protein or peptide is phosphorylated by these enzymes and such. They need to access it and have the recognition, and there the prolines and
very much.
the proline
So
to
there
type
are
2 structure can
in that other.
Yeah.
So that could be something that also got a bit stabilized upon from the binding. Yeah, that's and I do not know how much is known about the kinase that phosphorylate this protein. Is it known which kinase it is? Or
that I haven't looked into.
Yeah, that also could be interesting to figure out how they recognize each other and how the mechanism of phosphorylation is. So you're reading a lot about your study on phosphorylation of this staterine protein. I'm still left with a question. So in the saliva, is it known if it interacts with other proteins? Or it's more a function that it does through self-assembly and binding with calcium?
I think it's binding with calcium and like absorbing to the tooth enamel. And there, in this tooth enamel, there are a lot of different proteins present, and how if they affect staterine in some way, I don't know.
That's just because one of my main interests in the IDP field is also more this short-term linear disorder functional motif as modules for protein-protein interaction. I don't know, but maybe this is the case in which more of the function you can explain it through the mechanism of self-association and eventually interaction with the metabolite.
And I think this like C-terminal part with the glutamines and tyrosines enables more self-association and also then perhaps the interaction with other proteins as well.
Yeah, and there I think one of the next steps should be to go to try this interface operation constantly. Then maybe I give you with a couple more general questions so we can close it. It was a very nice discussion. In your case, because there are also challenges in studying phosphorylated proteins experimentally, because in your case, as I understood it, so we have a small IDP. I don't need to produce the phosphorylated version from the cell and purify it. So you can buy a peptide and then you purify it and you do the measurement.
Yes.
But let's see now you get contacted by someone, like the application that is a crazy biologist, and it's a very complicated disorder of protein in which they are actually interested in the phosphorylation. Could you suggest them some alternative to, because you are always in need, so you are always in need of going together with your model and your biophysical experiments. And then at the point, they will not be so willing to try to purify the protein for you. Could you suggest them some alternative in which they can still study with you the effect of the phosphorylation, what they could do without trying very hard to produce a chemical modified protein? If we think in the amino acid world, there is something we can do.
I know that sometimes people try to substitute it with just a negatively charged amino acid instead of the D or the E, depending on if it's serine or
yeah, because then mutation to produce a mutated protein probably is going to be easier than and then you work exactly with this, the concept of using phosphomimetic mutation. That of course again come with which one you would pick up to mimic, for example, your phosphoserine? Would you be more happy with the aspartate or the glutamate? Why?
I think the aspartate, that is the shorter side chain, and I think that would be more suitable for the serine one. It's more similar to the serine. But it would be interesting to do a computational study of how
it's the potential to reproduce the property of the phosphorylated one. I agree. And then you have also a nice model system to try that because it's not too big. And so since the serine is not the only residue that gets phosphorylated, because we have others, right? Do you think that phosphomimetic would be like aspartate and glutamate are going to be a good idea or a very bad idea for the other two residues that get phosphorylated?
I think like for the threonine, it can work as well with the glutamate.
Yes, something a bit more bulky.
Yeah. But then the others, it's harder.
Yeah, it's harder. And actually there are studies in which they really show that the glutamate or aspartate is not going to be good mimicking for the phosphotyrosine. Phosphotyrosine, sometimes big contribution is from the steric effect. It's not charges only. And it's difficult to adapt to think something else. And the last question, so you are at the end of your journey. It was a very productive one with a lot of new things for you to learn and some comfort zone in the beginning from, as I understood from your previous master. What would be, so now, what would be the most important, the one or two most important things to do on this system based on the knowledge you develop, if you have to choose what you would like to do next? And also if you can think outside the box and think that you can team up with collaborators, not only think about what you can do here in-house, but in general.
But one thing is what we discussed about statuine and the role of the tyrosines, and especially the tyrosine arginine interaction. I would like to see it experimentally as well, and then maybe try with the NMR methods. But then.
The NMR, you see it useful for what is to address which question, especially of the open question you have in mind.
So with NOE, for example, I guess you can see if residues come close, and that could be helpful.
Yeah. But to record NOE on an IDP is really
yeah.
nice.
Yeah, so it's challenging, and I think I would have to discuss with the experimentalist what is possible, and also like with this statuine self-associating and being sensitive to different conditions with aggregation and everything. It's tricky to work with, and perhaps the experiments we want to do aren't possible. But that is.
Yeah, or you get more creative talking together, and sometimes it can work out.
But another thing I think is interesting is to do more systematic studies of the effects of phosphorylation, since there maybe focus on just the number of phosphorylations or the charge pattern or something to understand that factor alone first.
And I think you have a really nice model to do that in a systematic way. So I think I'm happy with my question, and I think I took a little bit more than expected, but thank you so much.
Thank you.
It was a really nice discussion.
Okay, thank you very much. So I think
Can I come back now?
you're welcome to sit there. You don't have to. You can sort of, whatever you want. That's fine. I think we'll try to get Sarah on the line and see if we can. Can we get her on the screen, or should we use Ellen's for that? Yeah. So, I guess you could take Sarah and pin her. We don't need to see you.
No. You can try.
Du håller fortfarande din ledning ner. Jag tror bara pausade den.
Nej, men jag har gjort ett stopp på den sen.
Okej. Certainly. Ja. Kan vi göra den här lite. Okay, wonderful. Now you're in view, Sarah. So you're welcome to ask your questions.
Sure. So first of all, I'd just like to take a moment and congratulate you on a really beautifully written thesis. It was really a pleasure to read, and I was really impressed with not only the quality of the writing, but also the quality of the visualizations of your data. It was just incredibly clear and very easy to follow. So I guess I'll start off with some questions about conformational sampling. So you, in the introduction to your thesis, you had section 7 that included some discussion on assessing the quality of the sampling in the simulations. And you talked about 2 methods. You talked about block averaging, and you also talked about computing the correlation time. And you mentioned that these are very informative when you're looking at individual observables, but that these methods don't allow you to assess global sampling quality. So I was wondering if you can mention other methods that might be used to assess how well you're sampling the global conformational space accessible to the protein during the simulation. So that's the first question.
Yeah, so for example, this PCA plots of different replicates, comparing them, say something about the global conformational sampling, I think.
So could you think of any other methods? Because so PCA reduces the data just to these 2 dimensions, but of course we're talking about like incredibly complex conformational landscapes. Do you have any other like alternative metrics to assess conformational sampling?
Yeah, I'm trying to remember now what I read. No, it's right now it's blank. I don't know which paper I read it in. Because they are something you can calculate. I'm sorry, I can't remember right now.
Okay, so that's okay. We might come back to that later. You can take some time to think about it. I think you also had other ideas in your thesis on that. Okay, so we know that conformational sampling is a very challenging problem for IDPs, and you've demonstrated this very well in your thesis. And you used both coarse grain methods as well as all atom methods. And you made it clear that coarse grain offers an advantage in terms of the timescales that we can access. So let's say that you have like finite computational resources. And we know that coarse grained simulations will allow us to access longer timescales, whereas all atom simulations we expect to be more accurate. Taking into account this trade-off, in which cases would you feel it's appropriate now to use a coarse grained force field? And in which cases would you rather choose an all atom force field?
It really depends on what information you're after. Yeah, but a coarse grain can be a good start if you have the ability to expand into atomistic later. I think you always want to use the most simple model that can give you the information that you want, and in an with the acceptable accuracy. So like start simple and see where it gets you.
Okay, that makes sense. Okay, so then in the second published chapter, you mentioned that explicit treatment of salt improves the accuracy of the results, and you saw some issues due to the implicit treatment of salt. Do you think that this might be relevant to other coarse-grained models that have been used to simulate proteins as well?
Yes, it can be. I think that it's easy to overlook the effect of salt. Yeah, it is known that this Dubai-Hickel model, it is an approximation. It works better at low salt concentration, but when you're up there at 150 millimolar salt, more physiological conditions, it can be a bit more problematic. And if you would like to include like calciums or other divalent ions, the problems could appear even sooner.
Yep, that makes sense. And then a question to the fourth published chapter. So in that chapter, you compared two different force fields, amber and Charm 36M, and then you also had two different water models. So you observed differences in the ensembles in both cases, but I was wondering if you think the differences are more due to the differences in the protein force field, or more due to differences in the water model?
Yeah, that's a good question. And for the amber force field, just using it alone with its first recommended water model, what I've heard, it wouldn't produce good results. But with this, it would be more collapsed. But with this TIP4PD water model, it gets this more extended structure. So I've looked at them more as one entity and haven't really thought about what comes from the protein force field. Because with the amber force field, the parameters developed for the phosphorylated residues, they were also like developed for an earlier amber force field. So when you then made the changes for making it more better for IDPs, perhaps you affected the balance with the phosphorylated residues. So that is also something to consider and look over those parameters again. But I think work has started to be made on that.
So how much do the actual parameters for the phosphorylated residues differ between amber and charm in that case?
It's hard to say, because, like, for example, looking at the partial charges, they look different in just like normal serine, and then how it changes with the phosphoserine. It's I think it's hard to compare, so I haven't looked into that in detail.
Yeah, I think that I agree with you. It's hard to directly compare because there are many differences in multiple parameters, and narrowing it down just to one particular difference between the force fields probably is challenging. Okay, so then just a question to the final chapter. So you mentioned that net charge is not sufficient to predict the impact of phosphorylation on the global dimensions, and instead you have to account for the distribution of the phosphorylated and the positively charged residues. So I was wondering, like, if you could create a generalized model to describe the change in the global dimensions due to phosphorylation, what properties of the sequence would be necessary to include in that model?
That's a very good question, and that is, of course, something that we would like to try to do. And the first thing that I started thinking about is this: how to find a good measurement for like the charge pattern or specifically just the distribution of the phosphorylated residues and the positively charged residues. And I didn't really figure out any good measurement for that, how to like put it down into one parameter. But that I think would be of importance. But yeah,
Okay.
then depending on that distribution, then also other things can be more important. Like for statuine, it seemed like the tyrosines had a bigger role in it as well. So I think we really need to study more proteins and do it in more systematic ways as well. And also consider both longer and shorter peptides, because these are rather short, and then you can see a more direct effect. And then when you have longer proteins, you can have more compensatory effects, so they can cancel each other out. And perhaps there for longer proteins, and the net charge is a better predictor. So
Okay, interesting. Yeah, so that's that kind of relates to another question that I had about that chapter, which is how representative your set of, like, a chosen set of proteins is for the larger set of phosphorylated IDPs.
yeah, and I think it's only representative for rather short peptides, and perhaps if you look at like an individual region of a larger protein, but yeah, you cannot really generalize into the longer proteins.
Okay, then I want to ask you a question and ask you to speculate on something based on what you know from this set of phosphorylated IDPs. What do you think is a more important role for phosphorylation? Do you think it's more important that it alters the chain dimensions, or do you think it's more important that it would alter the local properties like secondary structure?
I think that it depends on the function of the protein as well. Because in some cases it's more important that the local structure is changing, and other cases it can be more important. Yeah. For interactions with other proteins, I think that what happens at the more local level can be of higher importance. Now I kind of lost my train of thought. Sorry, what was your question again?
So I wanted you to, this is really more speculation, but what do you think is more generally the role of phosphorylation? Do you think it's more important that it alters chain dimensions, or do you think it's more important that it alters local properties like secondary structure?
I think overall the local properties are more important. With the statuine and casein example, for example, yeah, these examples, it contributes to the ability of binding calcium and calcium phosphate, and then I think the global dimensions doesn't matter that much, and yeah, other IDPs involved in like recognition. Perhaps it can also be these local effects and the ability to hydrogen bond, or the charge itself. But also in a longer protein, perhaps the global conformations can be of higher importance, since it can then a different part of the chain can be more stretched out or more compact, and that can give access to different regions and different interaction sites. So I think that phosphorylation can have many different roles, and it's a really complex question.
Yeah, I agree. Okay, so just one last question. So let's say that you had access to unlimited computational resources and also resources to carry out experiments, and you had to choose another disordered protein to evaluate both coarse grained and all atom force fields. What would be your dream experiment?
That's a fun question. There are many interesting IDPs, but just we have been working a bit with this caseins as well, so beta casein and alpha casein, and they are challenging in many ways, and it would be interesting to dive more into them. They are similar to statuine in this way, with having the phosphorylated residues and self-association, but they are larger, so you can see different effects. So that is something I would like to work more with, but I know it's a big challenge.
Okay, thank you. So those are all my questions, and I'll pass it on to the next person who'd ask questions.
Thank you very much, Sarah. So I think we'll proceed with Con.
With me or with her? I didn't hear.
Con,
Oh,
you.
okay. Yeah. So I should also thank you for the nice thesis. Unlike the proteins that you studied, I found the thesis to be very well ordered. So that was nice and was really it had kind of a flow that was nice to see, and I don't always see this in the thesis. But of course, I have a few questions, and the first one is actually quite biological. What are the protein residues that get phosphorylated?
So serine is the most common one, followed by threonine, but then tyrosine can also be phosphorylated, and perhaps histidine as well. I'm not really sure of that. That is those I can think of.
You are absolutely correct, but in the thesis, I think you mentioned only serine and threonine.
Yeah.
Tyrosine is very, very important. But nice to see that you know it. Then in the thesis, you had a very short part, 3.8, that is about exchange repulsion, where you explain about all the intermolecular interactions. So where is the exchange? That's 3.8. Let's see which page it is. 15.
Yeah, I just thought of it kind of as a name for it. I haven't given it much thought.
Yes, there is exchange, but the exchange is a quantum mechanical term coming from the exchange of electrons to make the wave function antisymmetric. And what you don't mention it here, you just mention everything that has to do with repulsion. And it's not, of course.
Yeah, I didn't really dive into the quantum details of it. Yeah.
All right. Then you had a lot of the thesis devoted to phosphorylated IDPs, and you've done quite some work to estimate the force field and which one is better and worse. And I think if I understood it correctly that you would recommend to use amber with a tip for deep water model if I wanted to simulate an IDP. Is that right?
Yes, of these phosphorylated IDPs, it seemed to be the better choice here. And also this charm force field has been shown to have some problems for longer IDPs, also non-phosphorylated. Yeah, I would start with the amber out of these two, but then there are more force fields that I haven't explored that can be interesting.
If you have very limited time, where an experimental is chasing you and wanted you to simulate a folded protein now, which is phosphorylated in 1 or 2 residues, would you use the same force field?
This amber force field with this water model is aimed at intrinsically disordered proteins, and I expect that it might not work that well for globular proteins, but this charm 36M, I know when they developed it, they wanted it to be applicable to both folded proteins and intrinsically disordered. So there I would choose that force field. But then there are many other force fields available, and I haven't really looked into force fields for globular proteins. And of course, considering the effects of the phosphorylated residues in this charm force field that we saw, it's a good question of how well it works for the globular phosphorylated proteins. So perhaps we would need to explore another force field. So I would try to do some reading as fast as possible if they're really chasing me.
All right, last question. So let's say you're finished here and you go to a job interview, and it's not exactly dealing with the computer simulations of IDPs. And then the interviewer asks you if you could tell him what was the most important finding from your PhD. What would you answer?
So something that I take with me is that it's much more complicated than what you first think. And like when going into a PhD, I felt that experiments, they give the right results, that we can always trust experiments, but we can't. And we cannot always trust the simulations either. So it's hard to know what to trust and what to believe. And I think that is something really important, and that I've come to realize from my work, that it's much more complicated, and you really need to think carefully about stuff. And
so that's the most important thing that you have learned. But what is the most important thing that you have taught the world?
But here I think about the coarse grain model, and that we showed that this coarse grain model actually works well in many cases, and that you can simplify IDPs to that level and still get a rather good understanding of things. I think that is interesting and important.
All right, thanks a lot.
Thank you, Don. So, Per, go on, please.
Yes, I would also like to thank you for a very well-written thesis. So I would like to go back to this conformational sampling again, even if we have had it many times. I think it's nice that you made it very clear with your thorough investigations in the supporting information that you don't consider this as the all atom simulations as sampling everything, of course. But in some way you are relying that you are sampling enough that your the things that you are looking at are converged. So I see some formulation, for example, in paper 3, the phosphorylated with C36. More sampling is required, although it appears that the actual average values will not change drastically. So how do you know that? What do you base that on?
I based it on that we simulated it for a very long time, and the radius of duration, for example, looking at it how it changed over time, it was rather stable. So I think that extended it even more wouldn't change it that much. But I think that exploring it with more enhanced sampling techniques would be beneficial.
And why is that?
If we're stuck in like a local minima that we can't get out of, so we don't see the full conformational space.
Yeah,
and I think that just then continue running it for longer wouldn't help that much, but maybe try other techniques.
Yes, I agree very much. And then you have this, you have studied the same system with coarse grain model and all atom models. So that's, and you say you mentioned that this change in propensity of the beta structure is not captured by the coarse grain model, of course. But it seems like there is a gold mine of things that you could compare with those. For example, the conformational sampling, maybe. Can you comment on have you tried to compare those ensembles in some way?
No, I've compared like the calculated SAC spectra between the models and with the experimental data, and also with staturin and the phosphorylation, seeing how the radius of aeration changes upon phosphorylation. It expands in the atomistic simulations, but it slightly decreases in the coarse grain simulations. That was something I noticed.
But could you do more detailed analysis of? In principle, there should be a one-to-one correspondence between the conformations, right?
Yeah. You could probably try to look more at the center of mass of the beads compared to the center of masses of the residues, and compare the conformations like with the pair distribution and things like that, to see the global conformation, how it differs.
Yeah, I think there could be a lot of interesting things to do there. Just a suggestion for the future. So you mentioned this problem with that the charge of the phosphate is static with the -2. How would you try to improve that?
So first I would like to just keep it fixed, but to another value, and see how the changes are. But I think that having titrating charges might be interesting if the environment affects the charge.
Mm. Yeah, I guess you haven't tried that yet.
No, I haven't.
Yes, I would like just to comment on one detail on page 2481 in paper 1. You say that you conclude that the specific you get the bird maximum of 0.15 in the probability distribution. That the specific average conformation occurs during 15% of the simulation length. So for me, that's a contradiction with an average, a specific average conformation.
Yeah, specific average. It sounds a bit weird, but I think we mean that the conformation that is like the average conformation, but that
That has this sort of typical shape or something.
Yeah.
Because I wouldn't also say that it's 15%. I mean, I guess 15% is a probability density, right? So it's not really the probability for a certain.
No.
So it's just good to keep that it's not the same as the probability.
Yes.
I think I'll say with that.
Okay, thank you, Per. Haven't yet seen any emails with questions. We'll just check again. Still nothing. So is there anybody in the audience that would like to ask a question? No one in the audience? Okay. Then we will move on to the sort of the evaluation part of the proceedings. So the opponent and the examination committee will, and of course I, person, will join me in a different room, and Sarah should move to the other Zoom session, should have received a different link. So we will be seeing you there, Sarah, when we arrive upstairs. So we will go upstairs. And for the rest of you, you can sit tight in excitement for the result. So we will come back here to give the announcement. And I suppose it will be something like 20 minutes. I can't guarantee any time frames, but that's kind of a normal time. So there will be a sort of a break for everybody else while the committee is doing the hard work. So I will see you later on then.
So, in the name of the committee, I must say that we came very quickly to a decision that we are very happy to grant you with a PhD. I'm sorry that we were not slow enough so that you will be a little bit more relaxed, but we will learn. So next time there is somebody with having a small kid, we will discuss a little bit more. Congratulations.
Thank you. Yeah, Thank you.
Well done. With that, we finish the proceedings in this place. So we say goodbye to everybody online, and the others who are here will reconvene at Esther, which, for those who don't know where that is, just follow the crowd. Well, it's not exactly a crowd, I would say. But anyway, we will show. Goodbye to everybody online.